Hi everyone. In this video, we are going to be talking about the immune system, and we'll be focusing a little bit more in particular on the innate immune system. But to get us started, let's just think about some more general questions on this next slide. So, this first question here is asking about characteristics or features that could be found in the body as a whole, specific tissues or cells that would help prevent infection by some kind of pathogen. And a pathogen is something that can cause disease. So, bacteria, viruses, parasites, all of those would be examples of a pathogen. So, when we're thinking about this question, there are some sort of more physical barriers that we could think of as sort of the first line of defense. So, our skin should hopefully keep a lot of things out of our bodies. Um, Hair also does that, as well as does mucus that lines our mucous membranes. Digestive enzymes in our mouth, stomach acid, all of these are sort of physical characteristics that would hopefully prevent an infection from occurring in the first place. Um, we also have sort of more cellular or internal defenses, so different kinds of immune cells like white blood cells, B cells, T cells, other cells that are found in the immune system. And then we could also think of things within a cell itself. So we talked about lysosomes and peroxisomes, different organelles that might play a role in digesting foreign particles like a bacteria or a virus. So all of these defenses, well, not all of them, but many of these defenses require that the human body or the immune system be able to recognize a foreign particle. So, how does a cell or a component of the immune system know that something is foreign versus what it should tolerate? So, we would not want our immune system to attack our own cells because when this happens, that would be a characteristic of an autoimmune disorder. So, we want our immune system to only focus on foreign kinds of molecules, so something that could potentially be causing harm. And the way that our immune system does this is by recognizing um, self cells that are marked with a self marker versus cells that don't have that self marker. So there's a set of markers, and these are usually pieces of protein that are unique to all of us. So we all have different sets of these proteins. They're presented sort of on the outside of the cell by this complex of proteins called the major histocompatibility complex, or MHC. And this allows our own cells to be marked as being belonging to our body. So it's a way to identify cells that belong to us and that should be tolerated by the immune system. So a self marker will label the body cells as being okay and good to go, and those are tolerated by the immune system. And then conversely, if there's a molecule on the outside of a cell that our immune system doesn't recognize, this we would call an antigen. So this is something that will trigger a response in the immune system and be recognized as something foreign that the immune system will then try to deal with. So cells with a self marker should be ignored by the immune system essentially, whereas cells with antigens on the outside should be triggering some kind of immune response. Okay, so when we're thinking about these molecules that are being recognized as antigens, I want us to think about what kinds of molecules should these antigens be, or what should an antigen, what kind of molecule would be a good candidate to be an antigen? That's a better way of saying it. So we would want our antigens that are going to trigger an immune response to be specific to the pathogen. So it's really important that we're not triggering the immune system with markers that are found in our own body cells, right? So it should be something that we only are going to find in bacteria, or that we're only going to find in a virus, only going to find in a parasite. We wouldn't want our antigen to be something that could potentially be found on our own cells. We also would like the antigen to be required by the pathogen. So if it's something that's not needed by the pathogen, then the pathogen can just get rid of that molecule, right? So we want it to be something that's sort of more integral to the pathogen's life cycle or way of living. So these kinds of molecules are often called pathogen associated molecular patterns, or PAMPs. And this is just getting at the idea that they're pathogen associated, so they're sort of molecular motifs that we associate with disease-causing bugs, and they're molecular patterns. So these are often going to be proteins or carbohydrates or some other kind of small molecule. And this next slide here is just showing an example of different kinds of PAMPs that we might find in bacteria, viruses, parasites, and yeast. So, for example, lipoproteins are a combination of lipids and proteins that are found in bacteria, both gram-negative and gram-positive. We might target a flagellin, since almost no eukaryotic cells in our bodies have flagella, coat proteins on viruses, so that could be like a spike protein on the coronavirus. So you don't need to know these specific examples, but they're just sort of illustrating that there are um, particular kinds of molecules that we find in these different pathogens that could serve as targets for our immune system. Okay, so here this slide is outlining the sort of two arms of our immune system, and those two arms are innate immune system and adaptive immune system. And the innate immune system is always going to be coming into play first. It's the first line of defense. It's sort of always at the ready. The sort of defining characteristic is that it's very general or not specific to any particular pathogen. So it's going after anything that's identified as foreign, and it's not discriminating or like trying to direct its resources towards any particular thing. The innate immune system can be activated by the presence of antigens, and it's very quick to react. So it's quick to respond, but not specific. And then on the other hand, we also have the adaptive immune system, and this is going to take a lot longer to come into play. So it's going to need to be exposed to a particular pathogen, so a particular virus or a particular bacteria, and then after exposure, it'll take maybe a few days for this adaptive immune system to kick in. However, sort of to compensate for that slowness, it's a lot more powerful than innate immunity in combating an infection. So it's a lot more powerful, but it just takes a little longer. Um, if you've heard about antibodies, antibodies are a sort of key part of the adaptive immune system, and it's what allows for that specificity of the adaptive arm of the immune system. And as I said, much slower. So in the next few slides, we're going to be focusing on a few cells that are important in innate immunity. So these are all cells that are important for the innate immune system. So we'll start with mast cells. Oh, before we get into that, all of the cells that we're going to talk about are leukocytes. And leukocytes are just another name for white blood cells. And white blood cells are a sort of very general category of cells. It's like a lot of different kinds of cells can be a white blood cell. So we're going to be talking about three different kinds of white blood cells. Okay, so the first one that we're going to talk about are called mast cells. And mast cells are really critical in sort of coordinating an allergic response. And we think of allergies as being bad because a lot of times we're allergic to things that aren't dangerous. But allergies are also really important for immunity for things that actually we want to keep out of the body. Um, so mast cells have these little receptors on the outside, so that's shown here by these purple Y-shaped structures. And these receptors are able to bind to antigens. And if the receptor is activated by an antigen that it senses is foreign, then it's going to trigger this process where these sort of polka dot things are little compartments or sacs that have a lot of chemical messengers inside of them. And when the receptor is activated by an antigen, these little sacs will break open, and that process is called degranulation. But the compartments will break open, and all of these messengers will be released. And this is going to trigger a lot of different kinds of responses based on where the mast cell is. So if it's a mast cell in the skin, you might get hives. If it's a mast cell in your sort of respiratory tract, you might get coughing or sneezing. Um, if it's a mast cell in your gut, you might have intestinal distress. So the mast cell's response will depend on where in the body it's localized. But in general, one of the key molecules that it's going to release are these histamine molecules. So this is going to start the process of inflammation. So we're going to bring more blood to the area. There's going to be some swelling. We're going to be attracting other immune cells to the area. So it's going to set off a whole cascade of effects. But if this whole process is happening because you smelled some pollen, then you might take some drugs to eliminate this histamine response. So if you've ever taken antihistamines, that's trying to block that cascade from happening. Okay, so mast cells are involved with sort of coordinating an immune response, especially an inflammatory immune response. And they sort of are often found lining outside facing tissues, so skin and mouth and nose and respiratory tract, digestive tract, things like that. Our next immune cell is called a natural killer cell, and this is also a white blood cell. So, natural killer cells are named because they are naturals at killing other cells. And the way that they do this is by sort of sensing what kinds of molecules other cells have on their surface. So, natural killer cells are going around and sort of probing the cells around them to see whether they seem suspicious or not. So, a healthy cell should have a lot of self markers on the outside, proteins that signal to the natural killer cell that this cell is fine and healthy and normal. And if the natural killer cell senses those self markers, then it'll leave that cell alone. However, if a cell is infected with a virus and is maybe displaying viral proteins on its surface, or perhaps not displaying those normal self markers, then the natural killer cell will actually inject some chemicals into the cell and cause it to die, basically. So it's going around and trying to kill cells that seem not quite right. And there's a lot of sort of it's a complex balance of whether the natural killer cell is going to attack the target cell or not. And so there's you don't need to memorize this chart, but it's just getting at this sort of balance of does it have normal self markers or not? Does it have a lot of activating antigens that the natural killer cell is suspicious of or not? So different combinations of those kinds of outcomes can result in different responses from the natural killer cell. But the big picture is that natural killer cells are sort of roaming the body and looking for cells that aren't displaying the right kinds of proteins on their surface. And if they find suspicious ones, they will attack that target cell and cause it to die. Okay, and then finally, our last kind of cell is called a macrophage. And macrophage literally means big eater. And essentially, macrophages are really large cells and they have a lot of lysosomes on the inside of them. And macrophages will be going around and engulfing foreign particles. So they could engulf viruses, bacteria, but when they do engulf that cell, or sorry, when they do engulf that particle, it could be a cell, it could be not, it could be a virus. These lysosomes are going to come in and fuse with whatever was engulfed, and it's going to digest whatever was engulfed into little pieces. So we're basically doing the process of autophagy, but it's like a big cell sort of dedicated to that process on the inside. So I'm going to flip to this next slide, and it's just showing this whole process a little bit more up close. So this one is showing that bacteria are the things that we're engulfing. So it's showing a bacterium engulfed by a macrophage, and then it's going to be encased in this little vesicle. And then the next step is for lysosomes with a bunch of digestive enzymes to fuse with that and digest the bacterium. So we're just breaking that bacterium up into a lot of little bits. And then this third step is what I want to highlight on this page, but these little pieces that were digested from the bacterium are actually going to be displayed on the surface of the macrophage. So it's essentially taking what it just ate and putting the pieces of it out on the outside of its cell. So antigens from the digested bacterium are presented or displayed with different sorts of MHC molecules on the cell surface. So taking whatever it ate and putting it on the cell surface. And this is essentially going to help alert the rest of the immune system that, hey, we just found these particles. You should probably be on the lookout for things that look like this since I just had to digest this bacterium. So this will help communicate with the rest of the immune system. Okay, so those are the three cells that I wanted to go over for the innate immune system. And this next, sorry, I already talked about this. We were talking about sort of components of the innate immune system. So we talked about physical barriers, mechanical things like sneezing and coughing. I didn't mention this one, but also our resident microbiota. So the bacteria in our guts sort of take up space and resources that make it harder for foreign particles to make a home in our bodies. And then general immune responses are what we'll get into next. So inflammation and the inflammatory response are going to be other parts of the innate immune system that are just very general responses to foreign particles. Okay. So when we're thinking about infections, I think a lot of times the components of the inflammatory response are what we actually are thinking of. So here we're showing components of the inflammatory response. So you can see capillary widening, increased permeability, attraction of leukocytes or white blood cells, and systemic response. So essentially what we're trying to do with the inflammatory response is we're trying to increase the blood flow to an area. And increased blood flow is going to bring more fluid to an area, bring more immune cells to an area. It's just going to like help direct the body's resources to where they're needed to fight against this pathogen. But we experience this increased blood flow as heat. So if you've ever felt a mosquito bite or something and it felt really warm, that's due to the increased blood flow. And also redness is because of that increased blood flow. We also want to leave more space between the cells that line these blood vessels. So you can see here up at the top, the cells are sort of tightly packed together, whereas here we're leaving more space in between the cells. And this releases fluid into the tissues that makes it easier for our immune system to sort of move around in the area where they're needed. So you can see over here, these white blood cells are trying to squeeze out to where they're needed into the tissue where they're being called to. But also just extra fluid in general is going to make it easier for our immune system to fight the battle that they're trying to fight. But because we have this excess fluid from these leaky capillaries, that's going to lead to swelling because of that excess fluid. Um, and so that is sort of a side effect if you've ever had a swollen wound. Attraction of white blood cells is going to be what we see as pus. So if you've ever had anything with pus in it, they're basically dead white blood cells that have sort of lived their life and now they're being removed. And then systemic responses are things like fever or just general feeling bad. So if you've heard of the word malaise, that's kind of just like general gross feeling, maybe tiredness. So this is just sort of part of the whole body's response to this foreign pathogen as part of the inflammatory response. So a lot of times the symptoms of an infection are not necessarily the pathogen itself, but could be your own immune response trying to fight that pathogen.